To stimulate specific neurons deep inside the brain, we always faced a tough challenge, having to choose between invasive but accurate neural implants or non-invasive but less precise neural implants. Invasive electrodes require to insert electrodes directly into the brain tissue and get a nice communication with the brain. But over time, this immune reaction, scar tissue forms around the electrode, and the communication degrades. Minimally invasive implants allow you to place electrodes on the brain surface without penetrating it, which is much safer. But the electrical current spreads out in every direction and you lose all spatial resolution, which refers to the ability of the implant to focus on a localized area of the brain without affecting the surrounding tissues. Basically, you have to choose between precision or safety, but not both. There is also a second problem. To get better precision, you need smaller electrodes. But smaller electrodes have higher impedance that impedes the current flow, which means that you need high voltages to push enough current through them. And high voltages on traditional metal electrodes cause something called electroporation, where the electric field punches holes in cell membrane and also drives chemical reactions in the implant that produce toxic byproducts. So the smaller you go, the more dangerous each pulse becomes because of the higher voltage. However, the people from this paper try to address that by developing an implant that gets the best from both worlds. Trying to get around all of those trade-offs, they created an extremely thin, flexible sheet of parylene, a kind of plastic, and it is just 4 micrometers thin, with 60 tiny electrodes printed on it made of a material that we know and love called P.PSS. One of the characteristics of this material is that it stores a huge amount of electrical charge in a very small space, which means that it can deliver strong enough stimulation while keeping voltages below 1 volt, keeping the tissue safe from electroporation. Remember? This laid this implant under the dura mater, the protective layer that covers the brain, and let the tissue heal around it for 7 days. That subdural placement also solved another problem they ran into early on. In their initial experiments, they removed the dura entirely to lay the device directly on the brain surface. But dura removal caused cerebrospinal fluid to leak out onto the surface, and that fluid actually acted like a short circuit, stealing most of the current before it could even reach the neurons. By instead making a smaller incision and sliding the array underneath, they dramatically improved how reliably the current penetrated into the cortex. The key to achieving spatial resolution was using pairs of adjacent electrodes together, one pushing current in and the other pulling it back. This is called bipolar stimulation, and it delivers the electrical field between the two electrodes instead of letting it spread out. This is all cool, but how could they measure it? The implant was on top theoretically blocking the view. Well, to prove all of this actually worked, they used 3 methods. First, they mapped the electrical field by inserting a tiny glass probe through the sheet at different depths and positions and measuring the current directly. Second, they made the electrode sheet optically transparent and used 2-photon calcium imaging, watching genetically modified neurons literally glow green in real time as they fired, confirming the right cells and the right locations were being activated. Third, they ran 2 pharmacological experiments to confirm that the stimulation was reaching deep axonal structures and not the superficial dendrites, which was the case. It was reaching deep axonal structures. On top of that, the same device could record neural signals too, picking up individual neurons spikes as small as 60 microvolts from the surface. And after 200,000 stimulation pulses over 4 days, histology showed 0 visible tissue damage compared to the controls. So why does this matter? Think about visual prosthetics. To restore useful vision by stimulating the visual cortex, you need to activate very specific clusters of neurons with high spatial resolution. This paper is awesome because it shows that surface electrodes, done right, can match the precision of invasive electrodes with less tissue damage. And that is it for today. Thanks for watching. See you tomorrow and stay super healthy.